Hi, biology students. As you are preparing to finish this unit, I thought that it would be really helpful to kind of go over this graphic organizer and to review some really important things about the four major macromolecules, since this is what this graphic organizer entails. I will tell you, at any point during the year, you really should be able to name these four macromolecules for myself or for Mr. Suter. These are really the key to understanding a lot of things as we move forward. So, four macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And so if we talk about carbohydrates, since that was the first one that you kind of looked at when we were dealing with the videos that you watched by Mr. Anderson about carbohydrates. So if you remember, carbohydrates in simple terms are known as sugars. Okay, and so carbohydrates are known as sugars, and so the fancy term for that can be called a saccharide. And so that helps us know the monomer and the polymers for carbohydrates. We have monosaccharide and polysaccharide. The other thing that's really important to remember about a carbohydrate, besides the fact that it's a sugar, is that it's made up of carbon, hydrogen, and oxygen. And that carbon, hydrogen, and oxygen is always in a 1 to 2 to 1 ratio, which means that if I have something like 6 carbons, then I'm going to have 12 hydrogens and 6 oxygens. Like, let's say C6H12O6, which is glucose. And if you notice, that ends in ose. Most sugars that you will encounter will end in OSE. Glucose, lactose, sucrose, galactose, maltose, cellulose, they all end in ose. Now, there are some really large polymers like starch and glycogen that don't end in ose, but most other sugars do end in ose. And so glycogen is the way that animals store really big carbohydrates, and starch is the way that plants store really big carbohydrates. So if you think about like culinary class maybe, and they talk about a food pyramid or your plate, and they say about having a starch, they're usually talking about something like a pasta or a potato, and those are complex or polysaccharides. So they're big carbohydrates. And so the reason that they're complex carbohydrates is it takes longer to break those down, because the longer that it is, the more bonds that have been created between the monomers. And so each of those has to be broken down in your body. Where a simple sugar or something like a monosaccharide, it's going to break that down a lot quicker. When we talk about saccharides, the other important thing is to look at the shape. And so, yes, this is not the entire shape, but this is the main shape that will help you. We have two rings. You have a hexose or hexagonal ring, or you have a pentose sugar that is a pentagon-shaped ring. And if I were to actually look at this, there are other things that are coming off of this. There's oxygens and hydrogens and actually another carbon as well as here that makes that up. But this main shape is going to help to identify most of your carbohydrates. Now, technically, you could have a linear carbohydrate, but when it's put into water, that's when it makes the rings. And we know that most life takes place in water. So it makes sense that what we're going to see are these rings. So when you're looking for a carbohydrate, if there's a question and it's asking you, which macromolecule is it? For carbohydrates, you're looking for these shapes to help you, and you're also counting the carbons. Is it a 1 to 2 to 1 ratio and it has a shape like this? Then you should be 99% sure that it's going to be a carbohydrate. A lipid, that common name, are our fats. They also include steroids, waxes, oils as well, but fats is the main one for our lipids. And this also is only made up of carbon, hydrogen, and oxygen. The difference is in a lipid, there is way more carbon and hydrogen than there is oxygen. So when you're looking, if the ratio is 1 to 2 to 1, then it's a carbohydrate. But if there's a lot more carbon and hydrogen than oxygen, then it's going to be a lipid. The basic shape for a lipid, I'll show you one, is like a phospholipid where you have a polar phosphate head, and then you have these lipid tails, and they are your fatty acids, or we talk about a triglyceride. And I'm just going to do a basic rectangle to show that glycerol, and then you have these fatty acid tails that are coming off of it. When we deal with something like this, and I'm talking about the ratio, each one of these little points represents a carbon. And then coming off of the carbon are all these hydrogens. We call this fatty acid tail a hydrocarbon because it's made up of carbon and hydrogen. And so then there's 1 oxygen here, 2 oxygen here, and 3 oxygen there. So all of these little points are carbons and hydrogens. There's carbon and hydrogens over here and only 3 oxygens. So you can see this definitely isn't a 1 to 2 to 1 ratio. You're going to have a lot more carbon and hydrogen than you are oxygen. Plus these tails show you that it's going to be a lipid. So again, if you get, let's say, this long chain on the exam and it says, is this a carbohydrate, lipid, protein, or nucleic acid? It's made only of carbon, hydrogen, and oxygen. And there's way more carbon and hydrogen than there are oxygen in it, then it's going to be a lipid. As far as talking about the monomers, there's actually no true monomer for a lipid, but the best thing we can talk about and say is that they all have fatty acids and then some sort of headpiece on this that's polar, so like the glycerol or the phosphate head of the lipid. So I always put down fatty acids, even though it's not truly the monomer. And then a polymer of a lipid, we'd be talking about a fat, a steroid. You can specifically give an example like a triglyceride, which is what Mr. Suter built in that one example. So when we're talking about, what is unique about it or why it is unique, for carbohydrates, I always think about the ring shape as what is being unique for it, even though we're going to see that there's going to be a ring in something else, but that's because it contains a sugar. So like the ring shape is pretty unique. And then as far as the fats, they usually have long chains of fatty acids, which again are hydrocarbons. As we move down to talking about proteins and nucleic acids, so as we talk about proteins and nucleic acids, proteins also are made up of carbon, hydrogen, and oxygen. Oh man, so we've got carbon, hydrogen, oxygen, carbon, hydrogen, oxygen, carbon, hydrogen, oxygen, and actually nucleic acids are also carbon, hydrogen, and oxygen. But both proteins and nucleic acids also have nitrogen. So right then and there, if you're looking at a model on an exam and it only has carbon, hydrogen, and oxygen, you can cross out protein, you can cross out nucleic acid. It has to be a carbohydrate or a lipid, and then use those tools that I already taught you to figure out which one it is. If it has nitrogen, now you can cross out carbohydrate and lipid. It can't be those. So now you got a 50-50 shot with protein and nucleic acid. So what do you look for next? The next thing that you want to look for is phosphate. Okay, phosphate is a big deal. Phosphorus in nucleic acids are going to be the key to telling you, hey, it's a nucleic acid. Now, you might have thought, wait a second, Mrs. Smith, you just talked about this being a phosphate head on a phospholipid. It's true, there is phosphorus that's there on this one, but you're not going to find any nitrogen, so then you're not going to have a nucleic acid. So if you have nitrogen and you have phosphorus, you're going to be dealing with a nucleic acid. So I'm always going to look for the phosphorus for that. For a protein, the thing that will tell you that it's a protein is something called sulfur. The problem is that sulfur is not necessarily in every single piece of a protein that you're going to see. It's mainly in the entire protein, but not necessarily in all of the pieces or the monomers. Because when we deal with a protein, we usually talk about it in terms of its monomers. We talk about it in terms of its amino acids. And so when we're talking about an amino acid, that's because we have the setup of an amino acid that looks like this. This side over here is what we refer to as an amino group. This piece right here is the central carbon, and off of this is the really important part of an amino acid. It's R group. This is basically what's going to identify each of the amino acids and what's going to be really important for the next step. And then the last part over here, this is called a carboxylic acid. And you'll notice I was careful when I wrote. Why is it called an amino acid? Because it's an amino group, central carbon, carboxylic acid, an amino acid. The name is right there in that. And so a protein is this repeated until we get what's referred to as a polypeptide or a polypeptide chain, which is essentially a long line of this repeating pattern with a bunch of R groups sticking up the top. But if this is what your body produces, R group after R group after R group, and we're talking about amino acid, that's not easy to store. So if you have this really long chain and you have to fit it in a small place, what do you do with it? Well, you fold it. And so when we talk about a protein, it's actually going to be this series of twists and turns in order to get a protein. And so what's unique about a protein is the fact that it is folded. So the difference between a polypeptide and a true protein is the fact that you have that polypeptide that is then folded into the proper shape of a protein. Because for the protein, one of the most important things is that the form equals the function. So if you change the form, you change the function. And hopefully by the time you're watching this video, you've also started looking at enzymes. And enzymes are a specific type of protein. So if we denature the protein, if we've changed its form, we change its function. So I think about a candy bar. Okay, if you stick a candy bar in your hot car and you only leave it there for like 10 minutes and then you remember and you quick grab it and stick in the refrigerator, you might have changed the composition or the form just a little bit, but hopefully it'll go back close enough that it can still be considered a candy bar and still be used. It's like that with an enzyme. If you don't change the temperature all that much, it'll still work. But if you leave that candy bar all day in a hot car, it's melting, it's coming out of the package, it's gone, it's in your car seat, you're not getting that candy bar back. That's when you truly have denatured the enzyme or the protein, so it will no longer work. So that's our proteins. And then our nucleic acids, they're really important because we're dealing with DNA, we're dealing with RNA, and we're dealing with the energy of the cell. So we're talking about ATP as being a type of nucleic acid. The monomer of a nucleic acid is called a nucleotide. And so a nucleotide consists of having a ring. It's actually a sugar. So yes, here's that sugar that we talked about, the carbohydrate. There's one of them inside a nucleotide. So how do I tell then? It's like, wait a second, all of a sudden I saw the ring. Well, what did I say? This phosphorus is the most important part. Sticking off of the sugar is going to be a phosphate molecule. And then sticking off the other side is going to be a nitrogenous base. Now, they could give you a picture like this, or if they felt like being really tricky, they could give you a picture that looked more like this. And so when you look at this and you see those are the rings, if you look carefully though, each of the points is actually nitrogen because it's a nitrogenous ring or nitrogenous base that's there. And we'll talk about those more when we get to the DNA unit. But just in case, they could put this instead of putting the simpler one. But still, it's 3 parts, the phosphate, the sugar, and then the nitrogenous base. Repeating is how we start to get our molecule of DNA. And so if you go down the DNA line, this is where we're talking about all the different nucleotides. The nitrogenous bases are then joined in the center. And again, like I said, we'll get to that more when we get to the DNA unit. So if we look at our entire chart now, we've got the monomers and polymers, okay, for carbohydrates. Big important thing about them, of course, is the ratio of carbon, hydrogen, oxygen. Lipids, carbon, hydrogen, oxygen, more carbon and hydrogen than there is oxygen, and these long chains. If there's nitrogen in it, then we're talking about either a protein or nucleic acid. If it's got the phosphorus in it, then it's going to be a nucleic acid. If you see sulfur or it's folded or we've got this amino acid chain, then we know that it's a protein. So when we talk about the commonalities, there's a bunch of different things. Yes, we could say that they're all macromolecules, but I always like to say that they're all made of carbon, hydrogen, and oxygen. That's probably the biggest thing. In all living things, we're going to find carbon, we're going to find hydrogen, and we're going to find oxygen. So hope this was helpful going over your graphic organizer. Make sure that when you are reviewing for the exam, that you're either recreating this or making a table to kind of help yourself. Like if it was me, I might be setting up and doing a table where I listed all of the macromolecules. Then I listed their monomers, their polymers, maybe some examples, maybe their shape. And so then I make my own table here to review. It doesn't have to be another one of these. This is just the one that we used as a graphic organizer, but really restating and relooking at this information is going to be one of the best ways to help you study for this part. All right. Hope you have a great day.